Hello, everyone, and welcome to today's ASHI's webinar. My name is Ahmad Mustafa. I'm the Vice Chair of the ASHI Educational Committee, and I'm delighted to be your host today. Thank you for taking the time to join us, and we are excited to have Dr. Paula Arnold here to share her insight and expertise with us today. So just before we start, I have some housekeeping items to go through. So this webinar will be recorded and accessible on the ASHI for future reference. Following the webinar, you will receive an email with a survey link. Completing the survey will allow you to obtain CE credit for your participation. We would love you to participate in this webinar, so you can use the Q&A feature to send in your question anytime during the session. We will do our best to address as many as we can during the last 10 to 15 minutes. If we run out of time, be sure to include your name and email, and Dr. Arnold can follow up with you afterward. So it is my pleasure to introduce our speaker today, Dr. Paula Arnold. Dr. Arnold is an assistant member and the director of the HLA lab at St. Jude Children's Research Hospital, supporting the Department of Bone Marrow Transplant and Cellular Therapy. Paula's interests lie in several areas of hematopoietic stem cell transplantation, including HLA loss in leukemia relapse after HLA mismatched transplantation, donor-specific antibodies and desensitization, chimerism and prediction of relapse, and KIR genotyping and donor selection. Today, she will be presenting on HLA loss in relapsed leukemia following HLA mismatched hematopoietic stem cell transplant. Please join us and welcome Dr. Paula Arnold.
Thank you so much, Ahmed. And thank you to the committee for inviting me to give this talk. HLA loss is one of my favorite topics to talk about, and so I'm really glad to be here today. I don't know why slides are not. Okay. I have no conflicts to disclose, and most of the figures are created in this presentation are created with biorender.com. Just showing you a little courtyard at St. Jude that is entirely decorated with tiny little color mirrors. It's just a beautiful little place. And so let me put my pointer. Gosh. I don't know why I'm not. Okay. So I will talk about today in my talk. First, I'll present a case study of HLA loss after hematopoietic cell transplant. So I'll mention this as HCT often throughout the talk. I'll talk about background and importance of testing for HLA loss, laboratory methods for detection of HLA loss, and if I have time to get to it, I'll talk about our study here at St. Jude in pediatric leukemias after haplo-identical HCT. And so the clinical case is a 5-year-old Hispanic girl, and she was diagnosed with acute myeloid leukemia. And she was treated and was able to get into complete remission, or CR1. But she relapsed with 6% blast. And this was at an outside institution, and she was referred then to St. Jude for further treatment. And so we did her typing here at St. Jude. We typed her father, mother, and a full sibling. So she shares a haplotype with each parent, but she doesn't share any haplotypes with her sibling. And she also did not have any acceptable matched unrelated donors in the National Marrow Donor Program Registry. And so she was a candidate for a haplo transplant. And so she proceeded to haplo transplant using her father as the donor. And this is probably something everyone on this call already knows, but just to a brief reminder that HLA is located, these genes are located on this short arm of chromosome 6P. And HLA, they help the immune system distinguish foreign from self. And so in the transplant situation, the HLA becomes a foreign entity. And this can cause problems. It can cause graft versus host disease when donor T cells recognize this foreign entity and attack the recipient's healthy tissues. And it can also cause rejection in the other direction, where recipient T cells can mount a response to donor cells coming in, and can cause graft failure. So haploidentical transplant, and I'm showing it here with a father and a father to a child, because that is really the most common thing we do here is haplotransplants from a parent. And I'm showing the shared haplotype in green, outlined in green, and the mismatched haplotype outlined in red here. And, haplotransplant holds the promise of graft versus leukemia effect by virtue of this mismatched HLA on recipient leukemia cells. But we also know that with haplotransplant, we have to contend with detrimental effects of this recognition, being the most serious being graft versus host disease. And so back to our case, this patient did very well for about 1.5 years, but relapsed in the bone marrow, and with a MRD, or minimal residual disease, of about 30%. And the clinicians asked us, has the leukemia lost the HLA? And so what to do from here? And so just to introduce HLA loss. So it's long been known that HLA loss or downregulation is common in solid tumors, somewhere around 30 to 40%, depending on the type of tumor. And there's evidence of selective pressure from T cells. But alterations in HLA in patients with hematologic malignancies is pretty rare diagnosis. But we know this as HLA professionals, that this can cause mistypes if there's a high blast count. And so we can get like homozygosity. So we know it's an issue. But the very sort of first really methodical study was by Leukaemia and colleagues in the New England Journal of Medicine in 2009. And they looked at adults who had either AML or myelodysplastic syndrome. And they had a haplotransplant with donor lymphocyte infusion. So that's T cells from the donor. Those were either given with a stem cell graft or after the transplant for control of relapse. And 17 patients relapse out of this 43, and 5 of those 17, or 30% of the relapsers, were associated with HLA haplotype loss. And the mechanism of loss was by acquired uniparental disomy. And so this is obviously an oversimplified picture, but we're looking at chromosome 6. And so what they found were large areas of homozygosity where this portion of the short arm of chromosome 6 had been deleted, and then by homologous recombination or copying of the other chromosome by the replication machinery, this region became homozygous, so derived from one parent. So that's where we get acquired uniparental disomy. This results in copy-neutral loss of heterozygosity. So we still have 2 of chromosome 6. And, we would not detect this problem in a cytogenetics analysis. And so I kind of like to express haplotypes as blocks. And so essentially, if a patient is heterozygous or, in their leukemia and in their native DNA, then this process makes them homozygous at this region of the genome. You can also get focal deletions in individual gene loci. This is a little bit less studied, but I think really an opportunity exists to look further at these kinds of mutations after transplant. And so 6-PUPD has been shown to be a mechanism of post-transplant immune escape because donor T cells, you know, they have the ability to exert graft versus leukemia against these disparate HLA, but the recipient leukemia loses that mismatched HLA such that they're completely matched with the patient in that direction. It's completely matched with the donor in that direction. And so donor T cells no longer are able to exert a graft versus leukemia effect. And Vago and colleagues showed this very nicely in a cellular assay where they mixed T cells from the donor with the leukemic cells at diagnosis, and they got very good killing. But at relapse after HLA loss, they no longer had this effect. And so this sort of caused an explosion of studies that looked at HLA loss in different transplant situations in different kinds of patients. And we now know that HLA loss is common after relapse from haplo transplant, somewhere in the order of 25 to 50%, depending on the study. And so this is quite an important phenomenon to be assessing in the laboratory. We know that it happens in a variety of transplant situations, in adults, pediatrics, in different kinds of leukemias, AMLs, ALLs, myelodysplastic syndromes. We, in our lab, found it in a biphenotypic leukemia, myelofibrosis, Hodgkin's lymphoma, et cetera. So it's been observed in a wide variety of diseases, whether you have bone marrow or mobilized peripheral blood stem cells, different conditioning regimens, whether you have T replete, deplete, or selected grafts, and with T cell add back or not T cell add back. So in the form of prophylactic or preemptive donor lymphocyte infusions. And while different studies have found associations with different things, some of those things that they found were that it's associated with disease risk. So patients who have had multiple lines of therapy prior to transplant, those entering transplant with active disease. Some have found higher graft T cell dose to be associated, higher degree of HLA mismatch, occurrence of acute and chronic GVH, and also of late relapses. So after 6 months or after 6 years, after 6 months or after a year. And so what is the importance of detecting HLA loss? And the first thing is to avoid ineffective donor lymphocyte infusions because these cells will no longer mediate a graft versus leukemia effect. And the other is alternative donor selection. So leukemic cells without, so if you switch the donor to a donor that shares the other HLA haplotype, then the leukemic cells are fully HLA mismatched with this new donor. And at least theoretically, you may have a heightened GVL response. And so, let's see, how do we test for HLA loss? And so I'd like to talk about how we do it and also how some other laboratories have done it and have published on it. And so step one really is to confirm relapse. And so how do we confirm relapse? And I think those of us who are, stem cell labs, we're at least partially or maybe fully involved in post-transplant monitoring by chimerism. And while the emergence of host or recipient chimerism often indicates relapse, it's not a direct measure often of whether there are leukemic cells. It's definitely a good one, but to confirm relapse, we really need, well, my animations are not quite great, but so let's look on the left-hand side. So we can monitor the success of the transplant by morphology, and so HemePath can look at a bone marrow aspirate and count the blasts, and so that's one way. Cytogenetics, we can look at chromosomal translocations associated with the patient's leukemia, either directly at the chromosomes or with FISH, fluorescence in situ, typing or binding here of probes that can see these translocations. But these two technologies are less sensitive than some of the more advanced MRD type testing, or minimal residual disease testing. And so many labs do flow cytometry and look at tumor markers that they've identified in that patient's leukemia, and this can be a very sensitive technique. And then also molecular MRD, so we can look at fusion transcripts by RT-PCR or RNA-seq, so by next generation, and also we can look at mutations in the DNA. For example, FLT3-ITD is one in AML that can be followed by DNA, and then other genomics type methods to monitor disease recurrence. And so there's different types of methods to detect HLA loss. We can either select our leukemic blasts and then do some testing after that, or we can just take the population of relapsed cells and test those directly. And I'll try to talk about each one of these. And so one technique where you need to purify the blast either by size or by tumor markers, sort those. And this was done by Cressida Topolieri, published a paper in Luca Vago's lab, looking at SNP arrays. And these are just SNPs across chromosome 6. And what a SNP array measures is the heterozygosity or homozygosity. The log R ratio measures the copy number. And so at relapse, we have this region of the short arm where there's a region of homozygosity, but our copy number is still intact. And so this shows copy neutral loss of heterozygosity. However, some of the challenges that SNP arrays are not readily available technology for all laboratories. And so this is often not an option for some labs. But it definitely is very useful in kind of showing where that breakpoint is happening. And so what about methods that we are used to using in the HLA lab? We can enrich for blasts either with magnetic beads coated with tumor-specific antibodies, so antibodies that are binding to those markers that we know are in the patient's leukemia, or we can flow sort blasts with tumor-specific antibodies as well. So this is we isolate the DNA, and then we can put this DNA into any of our typing assays that we do normally in our lab. SSP, not that many labs use that anymore. Perhaps NGS, even RT-PCR, have seen that published as well, and SSO. And so the challenges of this is that purity is important. And so if you don't have good purity, you're going to have a mixture of your donor-derived cells. And also, you must have adequate tumor markers to follow, and there's always the chance that the leukemia could lose those markers, just like they lose HLA. The other is that a low level of relapse equals a low yield of cells and perhaps a low yield of DNA. So will you have, enough DNA to test? You can do flow cytometry for expression of HLA. And so actually, I think I skipped. Sorry. So this is an example of Shear and colleagues did this technology using magnetic beads coated with subset-specific antibodies for the leukemia. And they showed that the typing before transplant for the patient and recipient, both heterozygous, and then post-transplant, the blasts that relapse were just homozygous. So this is one way that this can be done in the lab. Okay, so back to flow cytometry. And so if we have HLA specific alleles that we have antibodies or flow antibodies for, then we can look at expression. And so Villalobos and colleagues published HLA loss, looking at three different patients at initial diagnosis. They expressed, the leukemia expressed HLA-A2. At relapse, patient 1 lost expression. Patient 3 didn't lose expression, and patient 2 didn't lose expression, but then got donor lymphocyte infusions and was shown to lose that expression. And so this is a great visual and technology if you have the right alleles represented in your patient and your donor. And some of the challenge are the commercial availability of antibodies to recipient-specific HLA alleles. A2 may not be in your patient or your donor, and you may not have antibodies that are adequate. Others is sort of gating on those tumor markers again. You may lose tumor marker expression. It only measures expression and not genomic HLA loss. And so we move to typing a mixed sample. And by mixed, I mean donor recipient cells are in there because this is a relapse after a haplo transplant or a HLA mismatched transplant. And so there are ways to do this. You just take your relapse from blood or bone marrow, isolate your DNA, and then put that into your typing assay. But the big challenge with this is that these methodologies are not designed for samples with more than 2 alleles per HLA locus. And so there is some manual analysis that needs to be done, and definitely quantitative validation, because these assays may not be sensitive enough to detect that third allele, and especially if you have really low relapse. So the question is, what is HLA loss versus I just can't detect that in my sample? And so these are things that the lab needs to really think about. There are a few kits out there that are designed for measuring HLA loss. This one is from GenDx, and this is one that we use in our lab for HLA loss. And so we just take our relapse sample, we isolate DNA, and we put that in a chimerism assay. And there are markers that are represented across the genome, and those are indels on the non-HLA chromosomes. And there's 10 HLA markers, 5 on HLA-A, 2 on C, and 3 on DP beta 1 that are target single nucleotide polymorphisms, or SNPs. If you're interested in validating this in your lab, this is a paper that describes the validation of this reagent, and it's a very good paper to, very good resource. And so just to kind of, explain how it works, and this we're starting from the upper left and going clockwise around. And so just as an example, if an insertion deletion, a recipient has the insertion, the donor doesn't, we add primers and probes. The probes have a reporter and a quencher in close proximity. And so the reporter can't give off fluorescence because it's being quenched. And so your polymerase is going to read through those, that probe if that sequence is there, and it won't obviously bind if the sequence is not there. When it reads through the probe, the quench or the reporter are released from each other, and you get fluorescence. But if you don't have that sequence, then you don't get any fluorescence. And so a positive reaction looks like this, and this is the number of cycles it takes to give off the fluorescent. CQ being the cycles, the quantitative cycles that it takes to cross that positive threshold. And there's math involved, and the readout really is, so classical relapse would be a relapse without HLA loss, is you would have a signal that's similar with your non-HLA markers as with your HLA markers. And an HLA loss relapse you would have relapse or host chimerism with your non-HLA markers, and then a drop or a lack of signal with your HLA markers. The big benefit of this is very sensitive. The detection level is very low. The quantitation, we lose a little bit of sensitivity on the quantitation. The non-HLA, because their indels are more sensitive, but the HLA markers are SNPs, and they vary in sensitivity depending on them. The disadvantage is that with only 10 markers, 2/3 of patient donor pairs don't have an informative HLA marker. And it's not accurate at high host chimerism, which we know to be a flaw of qPCR. But if we have really high host chimerism, so a high level relapse, we're really looking at the difference between non-HLA and HLA signal. And usually that's very clear. Okay, so if we don't have an informative HLA marker in our lab, we use just SSP, which, not a lot of labs use SSP anymore, but it's actually really useful for this type of testing. And we just get DNA from our relapse sample, and we look at, and this is of course a manual analysis, we look at the bands that should be there. If we see the recipient specific, so we have 3 alleles, if the HLA is not lost, and we see that band. If we have HLA lost, the recipient specific HLA is no longer there. And but it's really important again that we validate this sensitivity. And we did an extensive validation, and we found that 6% recipient chimerism. If HLA is there, we're going to see that band. And so the advantage is really quick and easy, but the disadvantage is that it's not as sensitive as qPCR. And so you have to have a certain level of relapse to test this. And then secondly is that SSP is being phased out. And so we have to think about what other methods we're going to use for HLA loss. And so maybe make sure I'm good on time. And so our clinicians are saying, has the patient lost HLA? And so we did our qPCR assay. And so this is day 600 post haplo, and this is the first transplant from dad. And our signal on our non-HLA markers is indicative of relapse, and our HLA signal is very similar. And what we're measuring here is the A01 that the patient has that her father does not have. So this is recipient specific. We also, why? We also used SSP for comparison's sake. And we're looking at, you can run whatever trays are informative. We're running a HLA-B tray and a DP beta-1 tray. And so we're looking at the patient-specific B8 and the DP 0402. And it may not be that clear in the presentation, but what you can see is these boxed wells, they're positive. There's a band there. And so this confirms that there's no HLA loss of a recipient specific. And so this patient then proceeded to haplotransplant with her mother. She was given donor lymphocyte infusions for low-level mixed chimerism, and these were increasing doses, multiple increasing doses of T cells in those infusions. But she relapsed in the bone marrow with about 1.5% MRD, so minimal residual disease. And so I'm showing her mother and their HLA in red, the haplotype that's mismatched. Sorry. Okay. And so we did qPCR on this sample. Because we can't do our other assay. We can't do SSP because it's not sensitive enough to detect this 1.5 to 2% of relapse. And so this is day 222 post Haplo-2. And you can see that we have a signal with our non-HLA and a lack of signal with our HLA. And so this is HLA loss. And we're measuring that A11 in the patient. That's different from the mother. And so the nice thing about this is that we detected this at a very low level relapse. And so if our physicians were thinking about doing a donor lymphocyte infusion to try to knock down the relapse, they would not do it here because we would have no benefit of with HLA loss, we have no benefit of graft versus leukemia and only the detrimental effects of graft versus disease. And so she got some chemo in hopes of getting that relapse, getting her in remission. But she ended up relapsing in the bone marrow with a frank relapse at 52% donor, and her fusion transcript for her disease was 1 in 10. And so they asked us to test it again because it was very low before, just to see is this still HLA loss? And so this is day 313 post Haplo-2. And we have in the open triangles, very high level of host chimerism, high level of relapse, but again, no signal with the HLA markers. And then if we look at the SSP, what we see, and I've put these in with yellow arrows, these are the wells that should be positive for the recipient specific HLA, but there's no band there. And so that confirms that there's HLA loss. And so the summary of this case is that the patient relapsed after haplotransplant from dad without HLA loss. So her leukemia had not lost recipient-specific HLA, but she did lose the haplotype inherited from her father after the relapse after her second haplotransplant from her mother. And sadly, this patient passed away from progressive disease, but perhaps detection of the HLA loss could help other relapsed patients. And so I'm pretty good on time, so I'll introduce. So we just published this retrospective study of our St. Jude cohort that we have been analyzing. And this is a retrospective study. This was 53 patients who had 60 total haploidentical transplants. Most used reduced toxicity conditioning. Most all used GCSF mobilized peripheral blood stem cells, and 63% had post-transplant donor lymphocyte infusions. These patients had confirmed relapse. If you look in the pie charts, diagnosis, AML, ALL, and some other acute leukemias, these patients were highly pretreated and high risk. Many had more than one transplant already. And so, and then their disease status at transplant based on complete remission. So the higher CR means more treatment, more relapses, or had active disease going into transplant. And so the vast majority of our patients were very, very highly pretreated. These are what they look like with the with our qPCR assay. We also tested some by SSP. That's not shown here, but you can kind of see you have different levels of relapse, but the same kind of pattern of classical relapse and HLA loss relapse. 15 of 60 of the relapses, or 25%, were associated with loss of recipient-specific HLA. And I'm only showing one piece of the data, but we saw very significant association of disease status at transplant in the occurrence of HLA loss relapses. And so patients that were in CR3 or CR4 or had active disease at transplant were at least 12-fold more likely to relapse with HLA loss. The outcome in our cohort was very poor in HLA, in our HLA lost patients. And so at 5 years, only 13% surviving. And that's upper left here. And then in the lower left here is all of our patients that relapsed with HLA loss. There's 15 of them. And it showing 2 patients that actually survived out of this group long term. And the interesting thing about those 2 patients, although it's anecdotal because we don't have the numbers, but the only 2 long-term survivors were transplanted with a donor mismatched for the other haplotype. So they had a transplant from dad. They lost HLA, recipient-specific A, inherited from their mother. They got a haplotransplant from their mother second and are alive today and relapse-free. And so, and several other groups have anecdotally noted this phenomenon as well. And so the conclusion from this is that HLA loss after haplotransplant is a common mechanism of immune escape in acute leukemias. And assessment of HLA loss at relapse is important for quick treatment decisions, primarily avoids ineffective donor lymphocyte infusions because, again, we have no benefit of graft versus leukemia, but all the risks of graft versus host disease. And secondly, informs selection of alternative haploidentical donors. So if your patient, is able to get to another transplant, the idea being that the alternative donor T cells are completely mismatched with the leukemia. And so, what's next, right? And really, what would be wonderful is if we had a chimerism assay that incorporated HLA loss and chimerism, that could be very part of the sort of testing that we do daily for chimerism. And so we're hoping we have an NGS assay, and there's a couple in the works, I think. There is a digital PCR assay as well, which is going to be very sensitive. We have not tried this in our lab. I'd be curious to know if anyone has. And so that's kind of what's coming. Oop, I don't know why my my presentation is lagging. And then I didn't really mention HLA expressions except for the flow cytometry assessment of HLA loss expression. But right now we're looking just at DNA in our lab, and we're looking at genomic loss. But we know that leukemias lose HLA expression, especially class 2 expression. And there's several studies on looking at this, and it's quite common. And so I would really love to look at RNA. By RNA-seq, we do RNA-seq here at St. Jude, but haven't ventured into that yet. But I think it's a very interesting way to go. So finally, I'd like to thank everyone who has helped with HLA loss, especially the HLA laboratories, the people that do the testing. They love to do HLA loss testing. Wink, wink. And also the guys and girls in transplanted cellular therapy who have really been supportive of looking at this, and also the biostats people who I would not be able to do anything without. And so thank you, and I will be glad to take questions.
Okay, thank you, Dr. Paul Arnold, for this fantastic presentation. And now we will move on to the Q&A portion of this webinar. I think we have received few questions, but let me start since I'm the moderator, so I will take this opportunity. So my question is, like most of the tests that you use or the method that you use has already been accredited. So I don't think you will need to do more accreditation, but how do you deal with proficiency testing? Like, does anyone ask you about what is the proper way to do PT, and how do you make sure that this test is actually working in your lab?
So this is a great question. So there's no PT at all for HLA loss. And so we do chimerism PT because the assay is chimerism based. But we have internal PT where we blind. So we have collected multiple samples, some that have HLA loss, some that don't. And we have someone in the lab blind everything, and they can't do any of the testing. And the rest of the lab does the testing, and then we report it. And then we look at what we know it should be. That's how we do RPT. And so I do know that, so there's an international workshop project, and we're participating in that. And I think we'll receive samples pretty soon. And that's Luca Vago and Katarina Fleischhauer are running that. And they're going to send samples, and we're going to test them. And I think the hope is from this, we can sort of standardize HLA loss testing a bit, and also perhaps see how to provide PT for this. So that's a great question, but there isn't one right now.
Thank you. And my other question, and then I will open for the floor for discussion here. So it seems that it is like a routine test for all haploidentical match. What about if there is a patient with one single locus or two single locus mismatch? Do you still do the same test? And how feasible is this assay for the detection of one locus mismatch?
Another great question. So I didn't show any literature on like, mismatched MUDs, or patients that, or match related that might only have one mismatch or something like that. But you can do that if you have an informative marker. It's published like about 2 to 10% of MUDs have HLA loss, so the percentage is less. And I think Vago and colleagues are coming out with another paper, a much bigger study, and so we should get some more accurate percentages for those. They're just not well studied. And so I do try to reach out to the transplant team and say, this patient relapsed from a MUD and they have a mismatch that we can test. Do you want us to test it? And so I'm trying to standardize with my group, that we always test when we have a mismatch, or we always order the test. We're not there yet, but I'm hoping to get there.
Thank you. So I think we go with in order here. So the first question is, in your experience, how does detection of HLA loss affect clinical management?
So for my interaction with the BMT group, if there's HLA loss, they're definitely not going to do a donor lymphocyte infusion, and that's usually at a low relapse level anyway, that they would try to give a DLI. So they're not going to give a DLI. And if the patient can get back into remission, they would choose a donor who has shares the other haplotype with the patient if they can. Sometimes we don't have those kinds of choices, but that's kind of how we look at it here.
Okay, thank you. I think the next question here it's from Dr. Kelly Hedgman. Paula, you're amazing. I think some of the digital PCR chimerism and the cell-free DNA panels have both HLA and non-HLA markers, although very few HLA is digital PCR. Although very few HLA is digital PCR an option for loss of heterozygosity?
Yes. So there is a kit that's out there that has more markers for HLA, but we have not assessed that kit. We test by NGS chimerism, just our general chimerism. We do NGS-based chimerism, and we are hoping that we'll get markers on 6P that we can just run with our regular chimerism. So if we have a relapse, then our clinicians can say, can you go back? And so we'll already have the DNA and we can test it with those primers, but not yet, not available yet.
Yeah, I think we have another question, but it seems that you already answered during the presentation, but I will also say it. So we don't have flow sorting in our lab. Does that mean we can't test for HLA loss?
Right. So there are several groups that have published on using magnetic selection. And so if you have markers that are useful for your patient to select, their leukemia markers with magnetic sorting, then it can be done. You just have to, it's always good to test your patient pre-transplant to have like a baseline of what that test result's going to be, and your donor separate, and then your mixed sample, because you may still have a little bit of mixture after that selection. And so you just want to make sure that you've kind of, crossed all your T's and dotted your I's on that one. Yeah.
Okay. Thank you. The next question here, as someone in the HLA lab, how do you keep track of all other ways of confirming relapse? Talking to other lab section, talking with Heme-Onc service, what is your practice?
So for me, we are a pretty small program and we sign out all the chimerism. And so I'll be honest that I kind of like stock the chimerism data, and I will like reach out to the bone marrow transplant. But we're trying to, and I also will have physicians reach out and say, Paula, because they know now, Paula, this patient's relapse, can you test for HLA loss? So that happens, but we would really love to get it as part of our protocols, and so that it just triggers when you have confirmed relapse, that it triggers that test. And so we're working on that to get it incorporated into our clinical trials.
Yeah. There is a technical question here. For SSP detection of HLA loss, do you run pre-transplant sample to identify recipient and donor-specific allele bands?
So we don't. We predict what the SSP bands are. So in the software, you can predict it, and we always make sure that we're seeing the bands that we should see for patient and donor. If we see spurious bands, then we will go back and test the patient and the donor separately, just to make sure we haven't, we're not getting something that is unusual.
Okay. And we have a question from Dr. Dennis Helmick. Do you still give donor lymphocyte infusion in these cases? Maybe it doesn't help with graft versus leukemia, but don't donor lymphocyte infusion have other benefit? This is more like a clinical question, but
Yeah.
I don't know if you will.
Right. Yeah. So we do not, if there's relapse with HLA loss, we do not give donor lymphocyte infusions. Typically, they're not going to be effective against the tumor, and so it's in our program they don't give donor lymphocyte infusions in that case.
And Mario is asking, is CAR T cell therapy also experience antigen loss? Can you comment if this patient was a candidate for CAR therapy?
Yes, so that's a great question. So we have some trials that are allo CAR T therapies, and we have been asked to test for HLA loss, but we have not seen any HLA loss in that scenario. But I will say that we have not tested that many. So I think it's a great question that it could happen, I think. But I
We
don't
have
know.
another question. Do you ever see a loss of DP in the 10 out of 10 matched donor BMT?
Right. So we have tested some matched unrelated donor that had just a DP mismatch, and we have not seen that in our hands, but we have also not tested that many, and other labs, other programs have seen in well matched unrelated donors HLA loss. It's, I think, probably rare, but we really don't know how common. Probably 2 to 10% is kind of what we know from the literature now. But again, the numbers are really small.
Excellent. And we have a financial question here. Do you have a CPT code for billing?
So, good question. So I think we use the chimer. I'm not that involved in the CPT part of it, but I think we use just a chimerism testing CPT code, because there isn't a specific HLA loss CPT code that I know
of. Another question from Dr. Hitchman is the percentage in chimerism always your trigger for checking LOH?
So chimerism is the thing that we follow the most here. We do a lot of chimerism. We test patients every week for, I think, 3 months, and then go to every 2 weeks. And so that's the thing we're following the most. So it triggers for me, but you really, in order to test HLA loss, I would have that chimerism number and some kind of confirmed relapse of leukemia. So MRD or morphology or cytogenetics, something that confirms that the leukemia is back, right? Because testing for HLA loss without leukemia is probably not going to be useful.
Okay, thanks. And we have a question here, and I might add a little bit to this question as well. So how do you validate magnetic sorting with antibodies to tumor markers? And my ad is, is there a specific antibody markers for each cancer, or is just like a common antibody that you use?
So that's a great question. I don't have a lot of experience with magnetic bead. We don't do that here. We do sorts. So I don't have experience with that validation but I think probably they're validating like the purity of you know how much of your marker and how much something else is there is what I'm guessing. But I really don't know. And so there's several papers. I think some used like myeloid markers for an AML, but exactly which markers I'm not sure. But I think, let's see. So I know this paper used subset specific antibodies that may be leukemia specific. I cannot remember. And then so this flow cytometry study, they gated on CD34 positive, CD13 positive, which was I believe myeloid based on the patient's leukemias. So that's why it's important to know what you're targeting in those in gating or sorting or magnetic bead type procedures.
Okay, great. We have a question from Manly Chen. Great talk, Paula. A quick question about SSP procedure. Do you use the regular PCR cycles or reduce the PCR cycle number a little in a hope for a better specificity?
So we just use the validated, the same PCR programs.
Okay, another question from Terry Harville. We assay buccal and blood with SSO and NGS with good success in detection. Okay, I think this is just like a comment.
So can you repeat that?
I think it's just like a comment here.
Okay.
Yeah, there. So we have another question from Dr. Hitchman. So it seems Dr. Hitchman is pretty active in this talk. Are there standard for LOH? Have you worked with the ASHI QAS on this? I think you answered this question again.
Yeah.
Yes.
No, there's no standards on LOH.
Would NGS testing for LOH require a confirmation of HLA loss? I don't think there is NGS for LOH right now, is there? I don't think so.
No, but it's coming.
Yeah.
And so the question was, sorry, could you repeat the question?
Would NGS testing for loss of for LOH require a confirmation of HLA loss?
Confirmation. I mean, if it's validated, no, I don't think so. I think the validation it's important because it's quantitative. And so you have to determine, what level is not detectable versus its HLA loss, right? So it's kind of a negative result. So you have to know when you no longer can detect it, even if it was there, which is very important, that quantitative validation in any of these technologies.
Okay, I think we are close to time. I think we answered, I would say, 95% of the questions. So I would thank everyone for attending today's ASHI webinar, and a special thank for Dr. Paula Arland for sharing her experience. Before we go, a few quick reminder. A recording of this session will be available on the ASHI Please take a moment to fill out the survey after the session. Your input will help us to improve on future plans for webinars, and it will also a way to obtain your CE credit. Don't forget to check out our upcoming webinars and other educational opportunities on the ASHE website calendar. Thank you again, and we hope to see you again in the next event. Have a great day, everyone.
Thank you.